Kevin's really helpfully set the scene there, and I'm just going to talk about one of the genetic changes, SOD1, and then use that to segue into some new exciting data that we've got on a marker that we can measure in the blood in people who are affected by these conditions to start to test drugs more quickly or to screen them. It wasn't quite 20 years ago today, but this paper came out a little bit later, but around about 20 years ago I was studying people who were asymptomatic and were carrying an SOD1 variant that was pretty likely to cause their condition. And that was really down to the vision of my PhD supervisor at the time, was Nigel Lee and Peter Anderson, and I used to go to Hammersmith to meet in people from Heathrow who'd flown in from Sweden from families and then take them up to, have to apologise for the Hammersmith, which isn't the nicest part of London, and then take them up to the hospital there to do a PET scan. And it taught me a huge amount about, and I really started noticing and listening to family stories, and it made me think very, very differently about the impact of these conditions. So that does seem a long time ago, and what we showed, this is just a couple of patients that you can see on the left there. There were some changes that look rather similar or early to the changes that we were seeing in the affected patients on the right. But we struggled to persuade the journal to allow us to include this, and there was a comment really about how, is it really very relevant to be studying people who don't have any symptoms really and genetic causes. So a lot's changed in 20 years. William Osler was regarded as being one of the sort of founders of modern medicine as the way we do it, and he wrote an article in 1880, so 140 years ago. He described a family in Vermont and the Farr family, and there's a book actually about this, which is a good read actually. It's very, very good documentation of the background. And this family carry an unpleasant, difficult variant within SOD1. And again, 30 years ago, that gene was discovered and linked to ALS, and that was really the start of this journey. And as Kevin's already told you, the most amazing development has happened. It's early steps. It's easy to sort of feel that there's still a lot to understand about this, and we're all a little bit nervous about wanting to believe it in many ways. Certainly the data are solid. It's just daring to believe that we might have entered this era, really, because the idea that we might be able to at least slow down the condition, even a small amount, reliably, has eluded us before now. But this is a paper in the New England Journal of Medicine that's telling you it's something regarded as very important by an American group and Biogen, who have come up with this molecule, tofersen, which binds to the SOD1 gene. And as Kevin pointed out, it's a small nuance, but it actually doesn't target the bad copy, the variant. It just generally reduces this protein, which is kind of interesting, really, that would both work, and it's obviously reducing the bad protein as much as, or more so than the good protein, we think, but that also doesn't seem to cause any adverse effects. So we're learning a huge amount just at a very basic level. And what you can see here on the slide is, in the middle there, as Kevin showed you, the change in people's worsening disability is fairly modest. And we've seen this in other studies, small effects, disappointing. We don't see much at 6 months. It tells us that we really can't expect to see clinical benefit in 6 months. We can't run trials over that period of time and make decisions. And unfortunately, the field is lagging a bit on that. So there wasn't a clinical benefit at that stage, but it seems to be getting more reliable as we go further out. But what was seen separately, and I'll point you to the panel in the bottom right, is that the people treated with the drug, their level in the blood and spinal fluid of something called neurofilament dropped very dramatically. I'm going to come back to what that means. And you can see that the blue line there, they're the people who didn't have the drug, and then it goes on to the dotted line, and then all of a sudden they go into the open label. That means everybody gets the drug, and their levels start to drop as well, telling you that it's a real effect. Now that's very important because that happened very, very quickly. And if it's happening quickly and ahead of changes in people's actually feeling that their disease is slowing down, or let alone possibly even feeling a bit better, that's very, very important, because it means that we have potentially a marker that we can get an early sign that there's likely to be later human benefit. So we can hang on and do the longer study because we've had that early signal. So what is this neurofilament? And I'm going to talk about neurofilament light chain. There are other neurofilaments are available, but I like light chain the best, and I think that's probably the way most of us are moving. Well, it's nothing specific to ALS. These are like the pieces of scaffolding on the side of a building, in the sense that they're part of the nerve cell, and when the nerve is damaged, they're released, and they're released into the spinal fluid, not surprisingly, around the brain and spinal cord. And they get into the bloodstream as well. And what's changed is not that we know about this. We knew about this back in 1993. It's not a new thing. And we knew they were raised, actually, in many diseases, including ALS and Alzheimer's and other neurodegenerative problems. But what's changed over 20 years is we can measure it in the blood. And that's made a huge difference, because it's difficult to take spinal fluid. It's still very important for our studies. And I'm grateful for people in the room who've donated samples to us. But it is nicer if we can do it from a blood test, and I'm sure they would agree. And the technology now allows us to pick that up sensitively. And what is the biggest barrier to developing therapies? Well, I'm not going to talk today about the things in grey here. We do want to make the diagnosis sooner. We do have a problem with that. That's complicated, and it's the way MND and FTD present. When we don't have a family history to sort of guide us. We want to understand why it's very variable. We know that it starts in different places, it moves at different rates. And the last one on that list is understanding who's at risk. And these are very important things, but I think that if I had to point to the one key thing, it's that I can't tell if someone's getting better if I give them a drug. If I give someone a medication, we don't expect them to feel better the next day, unless it's an antibiotic. But over time, we say to people, do you think it's working? And they don't know. And particularly if we're slowing the disease down, how do we know that the disease is slowing down? Because that might be a very important effect, even if it's not improving, if it's getting worse more slowly. But you can ask someone, they won't know. They don't know with Riluzole, for example, if their disease is slowing down, but we think it does from a trial. And we're not short of drugs to try, and we try them early on for safety. Those are called the phase 1 and 2 studies, basic safety studies and dosing studies. But to put them into the proper study where you compare a drug and a dummy pill, a placebo, that costs a huge amount of money and time, actually. It's going to take 12 to 18 months to do that study. That's for each patient. So that's going to be 2 to 3 years to get the answer, and then all the regulation after, it's years. And one of the problems is that we've tried a huge number of things over 30 years, and the results from those studies have been very disappointing. And so that this is all time to do, particularly ones in capitals. There were large studies that took 2-3 years to get the answer. Riluzole was positive, but a very small effect. Edaravone and the Amylyx compound, which is TUDCA and sodium phenylbutyrate, very small absolute differences, a bit like the differences we're seeing there with the SOD1 drug. Very small differences for the patients that are difficult for them to kind of quantify and to really feel any different or potentially feel warrants the treatment, particularly if it involves a lot of work, having intravenous treatment, for example, or treatment that may make them feel worse. Interleukin-2, we're still waiting for the data on that. It may be another one of these drugs that has a fairly statistical benefit clinically, but one that maybe patients won't be able to measure themselves. Again, we'll have to weigh up the complications of having to give these therapies and take them regularly. They'll have to weigh up what they feel the benefit is, because they can't measure it. So what do we want to solve? Well, we certainly want to improve that track record. We've been using a lot of mouse models and cell models. They're not humans, they're the best we have, and they do have some important insights. We've got a lot of pathways. You saw the whole range of pathways that we think are going wrong, And they vary really, a bit like fashion. You know, people are interested in one thing this year and then they're interested in another thing another year. And we have to try and make sense of that. And what we really lack is this predictor of human benefit. I think, you know, people living with MND and FTD say, you know, these trials are very long. And if you're living with ALS, a trial that's going to give you an answer in 2 to 3 years may be longer than you have. The cost of the trials we have to think about, because you know, we'll give our time as many hours as are on a day, but actually we have to persuade people to pay the staff to run the trials, and so cost is important. And actually we have to do placebo trials. That's a longer conversation, but there is no shortcut to that, because we know that you can have all sorts of effects that aren't actually due to the drug. And so asking someone to take a dummy pill for maybe 2 years, that might be the length of time they have left, is a very big ask. And I think if we could go to someone and say, yeah, but we are asking you to potentially be taking a dummy pill, but this drug, we've had much more of a hint that it works in humans. I think that would be a very different sort of conversation. And we do want to include a lot more centres geographically. So how are we going to do that? Well, this neurofilament light chain, NFL, that's what that stands for, is a protein that we can measure in the blood, and it has very high levels in those diseases marked in red there, and they've got an acronym there. That's HIV dementia, HAD. ALS, you know, CJD is a Creutzfeldt-Jakob disease, a very, very aggressive neurodegenerative process. You can see the one in blue there is MS, quite high in MS, but as you can see, as we move across, the levels get lower and lower as we go along, and we end up with Alzheimer's in one of the grey bars there, and Parkinson's disease is the last one. And what roughly it's telling you is that in an FTD, you can see is in the middle there in the blue. It's telling you really what we're seeing along that line on the x-axis horizontally is how long these diseases run for. Alzheimer's and Parkinson's are sort of 10 to 20 year processes sometimes, and FTD can be a 10-year disease. Whereas at the higher end, they're very much more aggressive diseases generally, not always, but it's telling you really that the level that you're measuring of this protein reflects how aggressive the process of neuronal damage is. And that's important because we want to have quite high levels so that we can maybe give a drug and we can know if it's falling. If the levels are already quite low, we might not be able to detect the difference. Now we know in ALS particularly that the level of neurofilament strongly predicts how fast your disease is moving. So if you have a very fast form of motor neuron disease, you will have a very high level of NFL, and a slower one you'll have a lower rate. And that's very important. That's been seen in many clinical studies based in clinics, and it's telling us really that, you know, if we believe that the people with slow disease and fast disease have the same disease, and there are lots of reasons to think they do, both when we examine them but also at the pathological level, that means that if you're lowering the neurofilament, that would seem to be a good thing to do. And also if you're giving a drug which is having a significant disease slowing effect, it would be expected to lower the neurofilament. We also know that what happens normally is important. We have to know what would happen to your neurofilament level if I don't give you a drug at all. I have to know what the normal is, the natural history, so that when we do give you a drug, we know if it's working. And the natural history is people keep the same level of neurofilament. You're seeing slightly slower patients on the blue graph there, then slightly faster in the green, and the fastest on the red. But each individual track there is relatively stable. So somebody's disease rate stays the same. And you know, we say that in the clinic, and we've done that for years, and it's a very helpful, important piece of information for patients. It's obviously nicer information for people whose disease is slower, but it's still important for people to understand their own trajectory. And actually this blood test, it's not necessarily something one can use individually, but in groups of patients, it strongly predicts both the rate of change, but we know it stays pretty stable. Now that's very important because it means that we could then start to give drugs. So just to remind you, it's not a marker of how advanced your disease is. It doesn't go up as the disease goes on. It's how fast you're moving. It's a speedometer. Now, Alex did some modelling of this from a large cohort of samples we collected called Ambrosia, which we did with Sheffield and London. And Alex modelled this, and what you can see on the right there is the number of people you need in a trial for what's called the power. So the power in a study is what's your ability to detect change. So to be sure that the change that you're seeing is meaningful, you need large groups of patients. And so more patients means more power, but obviously more patients, that's a bigger study, takes longer to recruit, takes longer to run, more cost. And also how quickly do you get an answer? And what you can see there is that you get an answer more quickly and for a lower number of patients with measuring neurofilament than you do looking for changes in disability rate. But actually we have some evidence from other conditions, really exciting evidence. And the paper on the left there is in HIV, and that goes right back to 2007 when the drugs were coming through that treat HIV. And what you can see in those graphs, you can see that the levels of something is falling quite quickly, a bit like they did in that SOD1 study I showed you. What's happening is when people get HIV, they start getting an HIV-related dementia process, they start getting neurodegeneration, their neurofilament is very, very high indeed. You give them antiretroviral HIV medication, neurofilament becomes almost undetectable, drops completely. So you switch off the process of that, and we can see that in those graphs there. And actually we can see some evidence in MS as well with a drug called fingolimod, which has some benefits in reducing the inflammation that causes MS. When you give that drug, you lower the neurofilament level and we see fewer MRI changes and fewer relapses. So we've got evidence that neurofilament is very closely linked to the speed of disease in people with ALS, MND, and we've got evidence from other diseases that when you lower it, that's a marker that you see ahead of clinical benefit. This is just summarising what happens. There's your neurofilament. It starts to rise in the early phase of the disease. We know that from genetic carrier studies. It's low, it's normal. We shouldn't expect to find any neurofilament in a healthy person. And then it starts to rise in 6 to 12 months before the onset. And then it settles out. And that period 3 there is where we would be looking to put drugs in people to see if it's lowering it. So the idea here is not to do studies like the past where we've looked at survival. That's the number 1 there on the left, where you've got a group of people, you measure who's still alive after 18 months. Very, very crude and doesn't always give you a simple answer actually. And then number 2 is the way we tend to do it over recent years. We look at the decline in disability. That's that ALS-FRS score. You can see there, bird's nest, all those different colours there are different trajectories of people in a trial. Some people falling very quickly, some people moving more slowly. That's what happens in a trial. You get lots of different people. And you give a drug, and what we'd like to see is drug X here is shifting the curve, making it move more slowly. Now that takes at least 12 months to show, and so that's a very long study as well. What we'd like to do is to measure the blood neurofilament level. This wouldn't be a study to prove that a drug works, but over 3 to 6 months, because that's what we think we can do it in a small group of patients, maybe 30 to 50 people, you could give a drug, and if drug Y here isn't making any difference to the neurofilament level, fine, let's park that one. That doesn't really seem to be of very much interest. Drug Z is lowering the neurofilament level. That doesn't mean it definitely works, but it's a really, really strong signal that it's more exciting. And so let's put that back into the proper study and say to someone, yeah, okay, we've got to do a proper trial of this drug, but it looks really exciting. We've had a signal. Let's do a proper placebo-controlled study for 12 months. A very different sort of prospect, really. And this is encapsulated by a project that's just kicking off, which we've called Experts ALS. Slightly worried about the term experts because I think it was Michael Gove said everyone's a bit fed up of experts. But it's a good acronym, and it captures experimental medicine. And what experimental medicine means is where you do studies in people. Now, it's not guinea pig studies, it's not safety studies checking whether something may have very severe side effects. It's giving drugs which have gone through basic safety testing and then seeing what happens to various measurements that you can make. So it's using really people as a way to get the signals rather than animal models or cell models. How does it work? Well, you take your drug on the left here. Now, how do we find those drugs? Well, initially we go to huge panels of what are called repurposed drugs, things you'll find in the prescribing book for all sorts of other things. So we're doing a study at the moment with terazosin. That's a drug for blood pressure or sometimes for an enlarged prostate. When you put it in a dish with motor neuron cells, which are going to get sick because they've been given some changes which produce changes similar to MND, when you give terazosin, it seems to protect those cells. That won't be because those cells have got big prostate or because they've got high blood pressure. It's what's called an off-target effect. It's something we didn't realise that terazosin does. And actually there's a whole raft of drugs, all sorts of strange drugs that you might think, wow, what they got to do with ALS, like metformin, that's a diabetes drug, or nifedipine, another blood pressure drug, or ropinirole, a drug for Parkinson's, and propranolol, actually, a beta blocker. These drugs are coming through in these screening panels as having effects to protect cells through some other pathway. Now, we know they're safe because they're used in humans for other reasons. That doesn't make us blasé, but it means we can be a little bit more confident about using them, and the regulation to use them is less. So we pick which are the best ones, and then we give them to a small group of people, and we get one of three outcomes. We might find that the neurofilament level goes up a bit. That's bad. We don't really want that. Or it doesn't really move. That's obviously the most likely outcome. It's not really changing anything. Both of those go into the low priority bin. We've tried them. We've quickly got rid of them. Probably it may take 3-4 months. But if we get one in the bottom there, the green one, it's actually lowering the neurofilament, then that goes rapidly into a proper study, a proper trial, and there are very many, well, a couple of platforms. MND Smart, you may have heard about, that's run out of Scotland, but with many centres around the country. And TRICALS is a European initiative. So the idea is to find drugs and prioritise them for the studies. And I think the information that's come out of the gene study, the SOD1 gene study, is really sort of turbocharged this, and that's why the National Institute of Health Research, the NHS's research funder, that's why they've funded this, and they have funded it. They've put several £1 million into doing this across the whole of the UK. And I think that's a good example of being led by a genetic study. Now, what's going on statistically is quite complicated, but we've got a lot of people working very hard on this, and we need modelers, and this is the Oxford supercomputer there. They have to, we're going to be measuring the neurofilament level every couple of weeks as samples come in from across the UK as people are on these different drugs. So it's going to be a real ramping up in our kind of activity. We'll have a kind of live readout, and we'll be saying, okay, we can see that this is a bit like the sort of NASDAQ, the share price. The NFL will be falling, or it's staying the same, or it's going up, making rapid decisions. So it's hopefully going to be quite a high throughput sort of system, this. But yeah, it's going to be quite a big change, I think. We're also going to be doing a lot of basic analysis and science studies on the samples. So we might get people within the study who don't sort of seem to do what the rest of the group do. So you can see in the top one there, although overall the levels are rising, we don't really like that drug, there was someone in that group who the levels seem to be falling for. They seem to be, in inverted commas, responding. So we'd like to kind of find out a bit more about why people might respond when others don't, and also when people aren't responding when the group is. And by doing all sorts of analyses and extra research there on the samples, we find out much more, and we hopefully, again, come up with better drug candidates. It needs a big piece of kit, and this is just to show you a couple of people in our group. Liz Gray and Ben Nazir are going to be managing this large piece of equipment, which is about £500,000 to run this assay for us. Now, going back to the genetic study, the obvious thing then to say is, well, I mentioned that the neurofilament level tends to rise just before you start to get symptoms in people who are carriers of SOD1 and actually other genetic causes as well. So you've got a marker which is telling you you're about 6 to 12 months away from symptoms. It's quite a big deal, really. And Michael Benatar will talk more about this, I'm sure, this afternoon. But he's been following people, and he thought, actually this is our first little chink of light, a window on maybe we could start to intervene earlier. And so what he said was, why don't we take a level here, that's that red line. When people's neurofilament level, we've been following them sometimes from their 40s, as their neurofilament level starts to rise, let's offer them the chance to get that drug tofersen, randomised to placebo, and see if it prevents or slows the onset of their condition. It's amazing, this is an amazing landmark study. Very, very difficult to set up, to organise, but there are a huge number of people already enrolled, and it's a real landmark study. So the idea, it's called the Atlas study, and the idea is that we see if we can delay the onset of symptoms using that neurofilament marker. Kevin touched on this case. I just want to mention it as well because, FUS is a rare cause. It's a very, very nasty form of genetic ALS. I can still remember when we saw one of the earliest cases in Oxford, and I was a registrar, and I sat in the clinic with Kevin. And it was new territory, really. This was, someone in their teens. And then we had another case. I'd just become a consultant. It was a young girl, and I looked after her on the ward. She died on her 19th birthday. And I was talking to her parents, and I thought, I'm not a paediatrician here. I'm not a doctor trained in looking after children. And this felt very, very different. And we teamed up with another couple of colleagues around the world, and we realised that this was a genetic form, and one of our pathologists was very smart in thinking about this gene and pointed at that. And since then, this has become much more ascertained, and this is a very exciting genetic approach, and it's had a rocky path. It was sort of almost on the brink of being given up on, but I think this particular case here, although the patient sadly still did die, they clearly plateaued. And this is a very aggressive condition. We probably have to treat it a bit earlier. And Neil Schneider is the lead author. Neil has talked about another case that he has, who's showing a much more promising result and possibly even improving. And again, what we're seeing is this early readout from the neurofilament. So again, the idea that the neurofilament may change quite quickly gives us really strong confidence that there's something to wait for. It's worth carrying on. And that has huge implications for being much more efficient with our trials. So last couple of slides. When does ALS begin? Well, John Rohrer lent me his version of this in FTD, and we'll see if he shows it this afternoon. I don't think John's here yet, is he? No. We all have these coloured sort of charts. They're important, really, to start thinking about things. And most of these will look similar to John. We can see disability rises, obviously, when people have symptoms. We can see neurofilament, the blue line, just starts to take off before symptoms. I think we can see there are behavioural and cognitive changes we can pick up quite early. We can pick up MRI changes and some of the brain function changes. More speculatively, I think there's a whole grey area, and Alex will touch on some of this in his next talk, where we're starting to see that there are changes that we can pick up in cells, both at things we can measure, but also more complicated cellular processes that are reflecting the fact, this thing that Kevin talked about, that why do you tolerate these genetic changes for so long, and then things eventually catch up. And that's not just simply brain reserve. We do have a huge reserve in the brain, but probably you're doing all sorts of things to compensate. And that may be a compensation that lasts your natural lifetime and you don't develop the disease. But knowing what those compensatory changes are is going to be the real meat, I think, of the next years ahead. And that's what we're focusing in on, really. Because those compensation changes may be very, very different to what we see in the disease. And we shouldn't look for the same things that we see in the disease state. We should be much more open-minded. And there's some very, very exciting but unpredictable things that have started to come through. Changes in people's lipids that aren't as simple as the story around heart disease, because we all know about cholesterol and heart disease. But we wouldn't normally have thought that these would be markers of anything in ALS and FTD, but they may be telling us, they may be what we call surrogates for how people are compensating. And if once we unlock and unravel that, then you can start to see who's most at risk. Even if if we can't necessarily immediately treat people and prevent the disease, if we could just say to someone, you're carrying these protective factors, it makes it much less likely that you're going to convert. That would be such important information. And I think we're starting to think about looking for the protective factors. Why doesn't someone get ALS or FTD rather than why does someone get it? Lots of complexity in that as well. We can see that we're starting to divide up what we mean by disease. I prefer the term, and I think other colleagues have told me, or people with lived experience, I think asymptomatic is a better term, because pre-symptomatic implies something's written in stone, and we know it isn't. But within this asymptomatic phase, we've got the prodromal features, where we might be getting some early behavioural or cognitive changes, or some motor changes, and then we've got a pre-manifest stage where we might have all sorts of biomarker changes. The complexity is getting more and more, really, is understanding when does disease really begin. And I went to a workshop last week. I'm very pleased to be invited to this. Michael Benatar in the middle there, you'll hear from later, and Terry Harman-Patterson stood next to him, organised a workshop really to start to think about risk reduction in people who are at risk. And all I would say about that is it was a very important exercise in saying what we don't know. Because we shouldn't be rushing in and telling people, you better not do this, or you better watch your lipids, or you better not join the military, or exercise. These are all things that are floating out there in the ether, but let's have the evidence before we start changing people's lifestyles. And this was enormously helpful to go through and say, what do we really know? What's really the evidence? Because it's very easy to sort of have an idea, and I've been very struck by athleticism over the years in our patients. But actually, the more I've gone on, the more I've kind of realised that it's more complicated than that, really. And there's a worry that, the easiest person to fool is yourself. The more you have an idea, the more you want it to be real. And we get these spurious correlations. This is a very famous one. This is a correlation, those two graphs there, the red and the green line, with apologies to people who might be colourblind, that they match each other. And one is the suicides by hanging and strangulation, and the other is the US spending on science, space and technology. So you can find all sorts of weird correlations. They don't mean that causes the other one. And you will get weird clusters. It's very difficult to imagine 3 soccer players, simultaneous ALS in the same village. It's very hard to not imagine there's something in the water or, something that's caused that. But actually, any epidemiologist will tell you all diseases will cluster. The really weird thing would be if you had a disease that didn't cluster, it was perfectly spaced, geographically and in time. There were no clusters whatsoever. That would be very, very strange. So we expect diseases to cluster, and we shouldn't see where the cluster is and, like the Texan sharpshooter, and then draw around it and say, look, there's I hit the target, there's a cluster. So we have to be very, very careful about appraising the evidence properly. And I think that we will be writing up the meeting from last week, but I can tell you that, I don't think there are any simple things we're going to be suggesting that people should be doing. I think it's a roadmap now for trying to understand what the relevance of these lipids and other things are, and metabolism and exercise and all these sorts of things might be. But there's not going to be a simple message that you need to stop doing this or start doing this.